We are going to talk about lab stuff. So I am a total lab rat. I love the lab. And this is actually taking me back to, these are my grad school. So when I left grad school to move on, I had to go clean out all my stuff and put it in my lab notebooks, and I had boxes and boxes of stuff and bottles of things. So all this is to say, I've done a lot of biochemistry research, and I'm more than happy to talk to any of you about any sort of lab things, experiments, all that. So most of my work involved recombinant protein work, and so I'm going to get to some of the things that are less recombinant, like a lot of you want to learn about CRISPR and things, and that's all happening inside of cells, which is awesome. But I also wanted to just give you a little overview of recombinant protein expression and purification and all that stuff, because I know that it comes up a lot in some of your papers. So one of the goals for today is to introduce you to some of the techniques you might come across in your papers, in addition to some of the techniques that are kind of just used more in the wild. So when we say recombinants, basically they're recombining pieces of DNA. So we get back to our central dot, we've got our DNA gene, it gets transcribed into messenger RNA, which gets translated into a protein. So the gene has instructions for making the protein. If we take the instructions for making a protein from any organism, so it would be a human protein, say, take the gene for the human protein, and now we want to say, okay, well, we want to study this protein. We want to, rather than have to study it with all this other stuff around in the cell, let's try to study it at the mechanistic level, get it really, really pure. And so let's get this into bacteria, and bacteria make a lot of this for me. So we take the gene for that protein and we stick it in a plasmid. And so plasmid is just like a circular piece of DNA and stick in bacteria. And there are bacteria that are really, really good at making lots of copies of the DNA or making lots of copies of the. And so when we do this, we recombine these pieces, this plasmid piece and this piece of the gene, and we stick that in bacteria. So we call that our transformation. And then the bacteria will then go and make the protein for you. And then you can purify it. So lysis, you break open the cells, you spin them down, you centrifuge them, you isolate the protein using chromatography. So we have these columns filled with these little beads that separate the protein based on their properties. And then we can do experiments with them. And what's really cool too is we're controlling what we put in the bacteria. So we can make changes to this sequence. So a lot of these times, like in your papers, they're going to study the disease version of proteins, right? So there's a mutation in the DNA that causes a change in the protein. And if we introduce that same mutation to the gene we stick in the bacteria, we can get protein B that is the same as can be found in those patients. And so this is often called site-directed mutagenesis, when we do the specific changes. And then we can go and see what effects that actually has on the protein. And so this is kind of the most minimalist approach, where you have in vitro. So we're outside the cells, and we're basically experimenting, trying to figure out how things work, like a mechanic. That's maybe like the least realistic stuff. And so a lot of times, if we want the most realistic, we want to think about in vivo. So in vivo in the living organism versus in vitro in glass. So typically, like we just think about like in a test tube or in that sort of thing. And then there's kind of this in between where you'll see things done in cells. And there's even not just cells, like flat cells on a plate, but they can also grow kind of mini organs that kind of mimic the organs. And so those are like organoids. People study things in tissue, so there's all sorts of different layers. And each layer, you're building more complexity. And as you're building complexity, it makes it harder to tell how that one protein itself works. But if you're just studying how this one protein itself works, like at the structure function level, then you're missing out key information potentially about how it actually works in the wild. But it's hard to tease apart the different components when you have this whole sea of things happening. So it's really important that we have experiments at each of these different levels, because each of them will tell you different things. And so different types of researchers will often focus on like a different level. And so they might be cell biologists doing more of this sort of thing, and more of the hardcore protein biochemists doing some of that in vitro work. And all of them are valuable, all of them are needed, and you'll see a lot of intercollaboratory research as well. Okay, so I just wanted to introduce you to this little scheme I'm introducing. And so I like to think about your cells or the cell's funny thing as kind of, or I guess not bacteria, they don't have nuclei. But DNA is stored in the nucleus. Then you have RNA transcription, they express the RNA copy. In us, it's going to get modified a little, and then the ribosome will translate it. And so it's kind of like a basic technology, and so that's why I drew it like that, and that's how I'm going to be representing some things. But of course, it really is at the level of those are actually sequences that are there. And the DNA, well, double-stranded DNA. One strand gets copied, and that strand has the RNA copy has the instructions for making the protein. And it has it written in these 3-letter chunks called codons. So 3 RNA letters is going to make 1 codon. Is going to spell 1 amino acid. And yeah, so I'm just kind of want to make sure we're all on the same page because I know some people have not taken really much biology, and so apologies if this is stuff that is really familiar with you, but hopefully this is a good review at least. Okay, and so then you have the translation step. The ribosome is actually going to go along, and this other RNA called your RNA is going to come. It's going to bring the corresponding amino acid. And so each of the codon, each of those 3 letters corresponds to 1 amino acid. And so hopefully some of these abbreviations are getting really familiar looking to you. Like, oh, that's lysine. Oh, lysine. Oh, glutamine. I know you guys. And you'll see that some of them have multiple codons that spell for them, whereas others don't. So sometimes what happens is that if you make a change to the code, so if there's mutation, and this is called mutation, or the mutation that you're introducing using that site-directed mutagenesis, you can change what actually gets made. But not always. So some mutations are actually what we call synonymous. So they change the spelling at the DNA level, but it still makes the same protein because of that redundancy and there's multiple codons that code for the same thing. But then we talk about missense mutations and nonsense mutations. So a missense is when it causes it to code for a different amino acid, and then a nonsense is when it puts a stop sign in there. And so the protein, it either if you make this truncated protein that is either non-functional or it gets degraded or something like that. You can also have insertions or deletions of larger stretches of DNA. So those are all sorts of terms that you might come across when you're looking at your papers. Sometimes you'll see a symbol that's delta, like a delta change. The delta symbol is often going to be used when you have a deletion. So if something's missing, you might see a delta. Okay, so you have this change in the gene that causes change in the protein. And that can cause some sort of phenotype. So some sort of thing that you actually see at the patient level, at the organism level. Genetics is basically where you go and you start with that phenotype and you figure out the genotype. Because researchers, they know there's this disease and now they're going to go and try to find what causes it. There's also reverse genetics where once you know what that mutation is, now you introduce that mutation and see what happens. So we're not really going to go into forward genetics because that's the sort of thing that the papers that come before your papers are doing. And so those are why when I was saying, we were trying to find papers for our project, I was saying it can't just be them finding the mutation. They have to actually be looking at how that mutation causes the phenotype. But how do people find out the mutation? So there is basically gene mapping, and so people can go and they can say, okay, well, grandma had it, grandpa had it, mom had it, and try to do these family pedigrees and track things down. And then go into even more detailed sequencing. There are things like genome-wide association studies, so you might have heard of GWAS studies, where basically they take a lot of data from a lot of sequences from a lot of people, and they try to find out which regions of the genome are correlated with what diseases, and then go in even deeper and look. And so DNA sequencing technologies have greatly improved over the past years. So you might hear, so kind of like traditional sequencing with Sanger sequencing. Now one of the main ones is aluminum, and so it does little tiny pieces of DNA, and it kind of like looks at the overlapping regions. There's also some newer methods that'll do longer reads. Okay, so we're going to talk more about reverse genetics. So you figured out what mutation is associated with this phenotype, and now you want to figure out the connection. So that's the key thing that we're going to be investigating. You're going to be investigating in your papers project. And so how do people do this? There's a number of different ways. So just remember we said that there were different kinds of levels of experimental systems. You have in vitro, you can have in vivo, you can have in cells and organoids and all these things. Well, we talked about the in vitro a little. So we can basically edit that gene in various ways, stick it in bacteria, purify that out, and then test what happens. But there are also changes. We can also make those changes if we want a more realistic idea of how this might actually be affecting the phenotype. What we can do is we can do things in the cells or in organisms. So there are gene editing techniques. So we'll talk in a minute about CRISPR, because I know that some of you are interested in that. And that you can make changes to the gene inside of the organism or inside of the cell. You can make changes that happen in a patient. You can remove the gene altogether. Any of those things are going to change the mRNA or prevent it from getting made. And then that's going to change the protein or the amount of the protein that gets made. If we're doing things like actually removing the gene, what we call that is knockout. So knockout is you're actually removing the gene or you're damaging the gene in such a way that no protein gets made. There are also methods of knockdown, where here it reversibly reduces how much of the protein gets made. So in grad school, I actually studied this protein argonauts. And so argonauts is used in RNA interference, and basically you give it a little piece of RNA and it goes and it finds the messenger RNA for the protein that you want to inhibit. And then it degrades that messenger RNA. So you're only getting rid of the temporary copies. So when we think back to our bakery analogy, it's like the chef makes copies of the recipe and gives those to his sous chefs or whatever to make the thing. If you get rid of those copies, you still have the original locked up in the nucleus. And so this allows you to lower the levels reversibly. And so we call that knockdown. And you might see terms like RNAi, RNA interference, shRNA, siRNA, all those various terms. There are also things that we can do to actually go after the protein. And so PROTACs, these are inducible degraders. Basically, you have multiple parts, and one part recognizes the protein, and then the other part will have it for degradation. And so you can actually degrade the protein rather than just degrading, or rather than degrading the mRNA or the gene. Okay, so let's talk. Because I'm because it is really kind of revolutionized a lot of fields from biochemistry, molecular biology, and actually patient care and medicine. So CRISPR actually comes from bacterial immune systems. So CRISPR stands for culture-regulated short column of ingredients. So basically they have these sequences in these arrays in their DNA. And when they get infected by something, they take a little piece of the virus that's infecting them, they take a little piece of the viral genome and they stick it in their own genome. And they're like, I'm putting you on my list. And now that virus comes, now they survive the infection and the virus comes back. Well, now they're like, oh, you're on my list, I recognize you, and I'm going to go and I'm going to degrade you. And the way that they actually go and degrade it is by taking that sequence that matches the virus sequence and actually using it to go and bind to that virus sequence and cut it. And so if we give them another piece of RNA that recognizes something that we want them to go attack, we can get them to go and cut that. So there are basically two components of a CRISPR-Cas system. You have the guide RNA, and so that would be, so in bacteria, they have the guide RNA that would say, okay, well, this guy was on my list, and this guy was on my list, and this guy was on my list. And those guys will bind to a Cas protein, and a Cas protein is what actually does things. And so different Cas proteins will do different things, but you can basically load them with the address of the place that you want to degrade. And so the guide plus the Cas will then go, and in bacteria, this is used to these proteins kind of go under surveillance. And they're like, okay, we're looking for you. And then what happens is that when they recognize the DNA with the matching sequence, they're going to bind. And then depending on the Cas protein, different things will happen. So the kind of conventional Cas9 is like the original one that was used as a tool. And it's kind of one force. It goes in and it's going to cut those strands. And then typically what's going to happen is that then protein just can't get made, and you defunctionalized it so much that no protein gets made, so that would be a knockout. But what can happen too is you can actually do gene editing, where you, instead of saying, okay, well, let's cut it, let's give it something to put in its place. And so you can do that, and you can get gene editing. Yeah, so the reason why though, so when you typically do it, if you don't put on something else, what happens is they try to kind of just stitch the ends together, and they're not very good at that. And so then you kind of get it knocked out. But if you do provide an alternative, well, now they have to do something called homology-directed repair. And it's not perfect, and it's not very efficient, and it only happens in some types of cells. And so there are then lots of innovation in CRISPR technology have been trying to get things to be more specific, because you want to make sure that you're not messing anywhere else in the genome, and to improve the efficiency of the system. And so there are different types of Cas proteins, both naturally occurring Cas proteins that people have found by exploring different unexplored microbes. So it's really important that we kind of do this basic research, understanding the variety of proteins out there in the wild, the variety of systems and organisms. So when Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for their work turning this kind of like CRISPR system into an actual tool that we use, they were studying the bacteriology systems. They weren't to figure out how they work. They were doing basic research. So they weren't trying to say, okay, well, let's go out and find a gene editing tool. Let's understand how this works. And now it turns into this thing. And so basic research is really important. And by doing more basic research, we can find more of these proteins and more of these systems that have proved to be very useful. And then scientists can also optimize them in the lab by doing like directed evolution and kind of introducing mutations to these proteins and seeing how they can affect things. In terms of so there's also various techniques that now will allow it to kind of, instead of cutting both strands, you cut one strand, so like a nickase, and then you can actually attach the Cas protein to different proteins that will modify the DNA. And so like you might hear of base editing and prime editing. So base editing is basically you go and you have it change like this specific letter. And prime editing is where you give it a template and have it like introduce a new sequence. And so you'll probably hear more about those terms in the coming years as they become more and more used. And yes, so one of the most exciting things, recently there was a report, and so maybe we'll talk about it a little more when we talk about the urea cycle, which is involved in protein metabolism. They don't express there. But they actually developed a gene therapy for a baby with a metabolic disorder, and delivered this CRISPR-based therapy and saved the baby's life. So really amazing stuff going on. So that's kind of, we made changes to the gene side of things. And then we could also out the protein in the wild. So those are kind of our two different sides of the equation that we're coming at a situation at. Often too, what you want to see, and this is going to play both in the work as well as doing things inside the cell, is that when you take that gene, not only do you make changes to the gene to introduce disease-associated mutations or something, you can also do things like add an extra bit on the end called an epitope tag. So we're saying an epitope, that is the thing that an antibody binds to. And so different antibodies bind to different epitopes. And so they might bind to viral epitopes. But also, their antibodies only recognize in their nose. The epitope is just like a really small part. And so we can actually just take a part that an antibody will recognize, and we can stick that part that the antibody recognizes, that epitope, we can stick that onto something else and trick the antibody into going and binding to it. And so we can stick these little epitope tags, these things like HA, flag, MIC, D5, KISS, we can stick, or this is like only an epitope, but we can stick those on the end of our protein, just by sticking the instructions for making them onto the end with the instructions for making our protein. And now we have this thing that we can use for various antibody-based methods that are actually going to allow us to detect the presence of that thing, or to purify the thing, all sorts of things like that. And so if you are taking Biochems 373, so the advanced biochem lab next semester, we're going to be doing a lot of protein purification. And so you will get to see a lot of cool stuff. But now let's talk about what happens when we make that change inside the cell or inside the organism. How do we actually go and see what effects there were? Because typically if you're working in cells, they're not going to be like, oh, my toe hurt. No, they're cells. They can't tell you anything. But we can go and we can look and see things. So doctors talk about like signs and symptoms, right? So symptoms are what the patient says, like complains of. I mean, not like they're complaining, but the things that they experience. The things that you can go and you can actually just see yourself without needing the patient to tell you. So these cells can give us signs. We can go and we can measure. We also know about this from like whether a gene was expressed, and that can be different things to different people. Like, so we have expression kind at multiple levels. So we have transcription. So we can go and use techniques like whole tube gypsy to see where that RNA polymerase is bound, where is it actually going and making copies. We can use techniques to measure those copies. So I'll talk more in a second. We can use like RT-qPCR, northern blot, microarray, RNA sequencing. We can measure how well copies of mRNAs are being translated. And so that would be like polygone profiling, ribosome sequencing. We can actually measure the levels of the proteins. And so mass spectrometry will measure like all the proteins. We can use a Western blot. We'll talk more about that in a minute. But that is allowing you to use those antibodies to detect specific proteins. And so yeah, so with like ChIP-seq, BRIC-seq, etc., what basically happens is that you can take proteins that are bound to the DNA. So maybe you have the RNA polymerase, and that's bound to the region of the gene that's fitting the genes that are getting transcribed in the RNA. So you know, like, oh, those genes are being expressed right now. And you can actually isolate the protein bound to the DNA and then sequence the DNA. In terms of measuring the RNA, so RNA sequencing is basically what it sounds like. You go and you sequence all the RNA. If you are interested in a specific RNA, there is a technique called RT-qPCR, reverse transcription quantitative PCR. This is similar to how like the test used to detect viruses, like the coronavirus. You say, okay, well, if you think about PCR, I don't know how familiar you all are with PCR, but with PCR, basically, polymerase chain reaction, so DNA is stranded. And what can happen is that you push it apart, so you heat them up. If that right, you can then direct a DNA polymerase to make DNA copies of a region you want, directing it to the sites you want to start at using short pieces of DNA called primers. And so you can use that to make copies of a specific sequence of DNA. And the cycle, does this in cycles. And so each cycle, what happens is you're going to double the amount that you get made. And if you don't start with any, then you can't make any. And so if the virus isn't there, there's no sequence to copy, and so you don't get anything made. If there's a lot of the virus, or there's a lot of the messenger RNA, you're going to have a lot to start with, and it's exponential. So if you start with a lot, you're going to make a lot more faster. If you are starting with a lower amount, it's going to take you longer. And so by then measuring the copies that get made, such as with fluorescent dyes, you can then see how much was there originally, based on how long it took, how many cycles it took for you to get to a high level. Okay, in terms of translation, so polysome profiling is a technique that can actually go and look at how many ribosomes, so the things that actually make the protein, how many of them are on each of the transcripts. And I can tell you about like which transcripts, which messenger RNAs, which proteins are getting made more. And ribosome profiling, where you actually see where the ribosomes are. And so I actually did a lot of this in my postdoc. If anybody is interested in ribosome profiling or polysome profiling, I can tell you more. Now let's talk about how we measure the actual proteins, because this is something that a lot of you, some expressed interest in and some So proteomics, so we use omics, that's like big data, lots of data, trying to study like a whole thing of things. So like a proteome is all the proteins that are in a cell at the moment or something like that. And so mass spectrometry, basically what it does is it will take things and it will make them charge, and then it will send those charged pieces through a device and then measure the mass to charge ratio of them. And so when I say things, so mass spectrometry can be used for like small molecules, but it can also be used for proteins, for proteomics. You take a protein that results are really big, but if you break it up, you get peptides, and then you make those peptides charged, and then you fragment them even further. You get these signals, and then you compare those to a database to figure out what protein was actually there. Typically, this is going to be after some sort of chromatography. So typically with proteins, you're going to be dealing with some liquid chromatography. And there are also like small molecules. You can do things like GC, gas chromatography. Okay, so now let's talk about those Western blots and other blots. So when we talk about electrophoresis, so we talked about electrophoresis the other day, talking about how like that stellar stuff. But typically the electrophoresis we're doing in a biochemistry lab looks a little more like this. The basic idea is that you have a gel mesh, and you have a plus charge at the bottom and then a minus charge at the top, or if you're doing it like this, you have a horizontal. So for like protein, you do SDS page gels. With DNA, you might do an agarose gel with it somewhere like this. And the charged molecules are then going to move through the gel, but the bigger ones are going to move slower because they're getting tangled up more. With proteins, we're actually doing this thing called SDS page. So sodium and methyl sulfate page, polyacrylamide gel electrophoresis. And here, remember that things have to be charged. When we talk about proteins have different charges, different proteins have different charges, and also they're kind of pH and all this stuff. And they have these different shapes. If we want to separate based on their length, how long that chain is before it pulls it up, without needing to worry about that pulled up shape interfering with its movement. Well, then we need to denature this protein, unfold it, and we also need to give them all a uniform charge. And so this SDS is a detergent, so it's like an artificial soap, and it's got this long non-polar part that's going to glob onto those hydrophobic parts of the protein and keep the protein soluble and keep it unfolded. And it's negatively charged head, which is then going to allow you to have this negative charge that's going to send the proteins through the gel thanks to the electrical gradient, and the proteins will travel through the gel. Because they're unfolded and they're negatively charged, they're very travelable through the gel at a rate that's proportional to how many amino acids they have. And now you go until like you have this tracking line that you use to see when the protein's basically about to come out. The smallest protein, where the smaller things down here, the bigger things up here. When we stop it, well, now we have this protein in the gel. And what we can do is a couple things. And so we can go and we can just stain it. And so we can stain the gel with just like a blue protein, a Kamasi stain, which will just stain all the proteins, and that'll allow you to see what's in there. And it'll allow you to see all the proteins that are in there. So you can tell how pure your protein is, maybe. You can tell how many proteins there are. You can tell what size the proteins are, how much in general, but you can't tell what those specific proteins are. If you want to tell what specifically they are, you're going to need to use the antibody-based method. Remember, the antibodies will recognize specific things, and you can have antibodies that recognize specific proteins. In a Western blot, so you don't want to send those antibodies traveling into the gel, but you can send them against a member. So you take the protein, so you have this protein in a gel, And now you're going to take them out of the gel and onto a membrane. Once it's on the membrane, now you're going to set after some blocking and stuff so that the antibodies don't just stick anywhere. You send in antibodies that have that will recognize the protein. And then antibodies that recognize those antibodies and that will be detectable. So antibodies, they have these kind of generic parts and these specific parts, and these specific parts are specific for the thing that you're looking for. And so typically what happens when you're using the antibody-based method is that you have an antibody called primary antibody that is going to recognize your protein specifically. And then you have a secondary antibody that will be bound to something that is detectable. So it might be a fluorescent molecule, or it might be an enzyme that you can then give a substrate, such as like HRP and stuff, that will then go and make a signal that you can see. And so the secondary antibody binds to the primary antibody, and that allows us to detect whether the antibody was bound to the protein, which then allows you to see if the protein was there. So your protein goes down. If you do chemistry, you see all the proteins, and that'll tell you, okay, the things on top are bigger, things on bottom are smaller. But if you're doing the Western probing, then you only see specific proteins. There's something similar called an ELISA that'll do things in a dish. This is just one type of what we call of a blot. And so Western blotting is, you often see it with a capital W, but it really should be a lowercase w. It is a kind of pun on the Southern blot. So Southern blot was the original blot, and it's named for a guy named with the last name of Southern. So that one actually is a primary noun. But then other people were like, okay, that's kind of fun. Let me name mine in direction too. So a Southern blot is looking for DNA with DNA. You also have Northern blots, which look for RNA with DNA. You have all sorts of different types of blots. Some of these are going to use antibodies. So antibodies are really great for recognizing proteins, but we also often are going to use, just because taking advantage of the sequence, the base complementarity between DNA bases and RNA bases and all that, we can use radio-labeled probes that will then go and bind to something. And so we can actually use radioactive phosphorus to replace the normal, we replace the phosphate with like a radioactive version, and that'll allow us to track it. You can also use antibodies to do a co-IP or precipitation. Precipitation, where you basically take an antibody that's bound to a bead and use that antibody to pull out the protein and see what it was bound to, say, using a Western blot. So some of you might see something like that. Also, there are things like MSAs that can measure binding, and so I just wanted to put this in here because somebody was interested, but now we're running out of time. But I do have these slides up there. I'm happy to stay and talk to anybody. I just don't want to keep you from your next class. Yeah, if there's anything else you want to learn about too, I'm happy to talk to you or send this for you to go ahead. If none of this is interesting to you, you're not responsible for any of the content, so this was just to kind of introduce you all. Have a good one.
Here's just a little more that I added after class because we ran out of time, of course, and there were still topics students wanted information about. So remember that the epitope is the thing that the antibody recognizes, and when we talk about antibodies, we often use a term called immuno. And so something, if it starts with like an immunoblot, it's using antibodies to detect something. If we talk about co-immunoprecipitation, we're using antibodies to pull out a protein of interest and see what it's bound to. And if we want to look at, like, for a specific thing that it's bound to, we can use a Western blot. But then we have to know what we want to look for, because we're going to have to use antibodies that recognize the second thing in order to figure out what was there. There are other types of pull downs that don't use antibodies. So say you have a his tag, maybe you can have a nickel beads to pull down a protein of interest. And then you do a Western blot if you want to look for specific things, or if you want to see everything that's there, you can do things like mass spectrometry, where you then cut up the protein into those pieces, you measure the mass to charge ratio of the pieces, and you compare the signal from those two data banks to tell you what proteins were actually there and present and bound. So you'll see like an input, that's everything you put in, and then the pull down is the stuff that actually came out with your protein when you pulled it down. So everything goes in, your protein sticks to the beads, the stuff that's stuck to your protein sticks to the beads because it's stuck to your protein. Everything else washes off, and then you see what is your stuck to your protein. Another way to see what gets stuck to your protein is a technique called a gel shift, or an EMSA, so an electrophoretic mobility shift assay. And often this is done to look at the binding of DNA or RNA to protein. You can also do a protein to protein, things like that. The key thing is that you're going to use gel electrophoresis, but unlike an SDS page, or unlike a lot of the types of gels that we use, these are going to be non-denaturing, which means we don't unfold the proteins, we don't unfold the molecules, we don't separate the strands of DNA, we don't unfold the DNA or RNA, and none of that stuff. Instead, we want to keep things as native in their native state, so basically how they are originally. And this is going to allow us to see if things were interacting. And so what happens is, say you have a labeled piece of DNA or RNA, and you want to see if it binds a protein. You mix the two together, and if the DNA or RNA is just by itself, it's going to be smaller than if it's bound to the protein. And so when it's bound to the protein, it's going to shift up in the gel. And so you're going to see it higher up in the gel because it's now a larger size because it's bound onto that protein. And then you can go and you can look and see how much, based on how much they like each other, how strong the affinity is, it'll take more or less of the DNA, or RNA. So if they like each other a lot, it'll shift really quickly. But if they don't like each other as much, you might have to have a lot of the DNA in order to see the signal from the DNA start to go up. But if they don't like each other that much, you might need to see a lot. So if they have a high affinity for one another, then you don't need much of the protein in order for all of the DNA to be bound. So the shift will start early in the protein concentration gradient. But if they have a lower affinity, then you're going to have it so that you're going to add a lot of protein before you can kind of get the DNA to come. And so that's just one method. That's kind of semi-quantitative, depending on how you do it. But if you really want to be quantitative, then there are other methods that you can use to measure binding affinity, and these include things like surface plasmon resonance, or SPR, biolayer interferometry, isothermal titration calorimetry, so that would be BLI, ITC, microscale thermophoresis, or MST, fluorescence polarization, dot plots, EMSA, and ELISA. And so there's lots of different methods for studying binding affinity. Some of them, like SPR and BLI, these are really good for measuring binding kinetics, so on rates and off rates, as opposed to just equilibrium binding. Another way that you can measure binding is by taking advantage of fluorescence. And so when we talk about fluorescence, basically a molecule absorbs a wavelength of one light and then it emits a wavelength of a different light. And so light is just little packets of energy traveling in waves, and different molecules will absorb different wavelengths of light because there's corresponds to different differences in energy between the electron energy states. Bottom line is that different molecules absorb different wavelengths of light and emit different wavelengths of light. I mean, not all molecules fluoresce, but some of them do. And some of them will actually do this thing called fret, where they will absorb what would have been given off as light. So remember that light is just little packets of energy traveling in waves, and so some molecules will absorb one wavelength of light and let off another wavelength of light, but another molecule might actually absorb that wavelength of light that was given off. And if the molecules are close enough, then the second molecule can actually steal what would have been given off as light, and so you don't see the light that would have been given off by the first thing. Instead, you only see the light that's given off by the second thing, but that only happens if they're close together. And so that gives us this concept of fret, where we can use to see if molecules are close together or if they're far apart. Something similar is quench fluorescence, where the second molecule, it'll absorb the light, or they would have been light, the energy that would have been given off as light by the first molecule, it'll absorb that, but it won't give off light. So you'll see that the light kind of disappears that you would have expected, and that could be used to see if molecules are close together. And there's something very similar called Brett, but more often you're going to see fret. And these sorts of assays can be done in vitro with purified proteins, or you can actually do some of these things in cells. If you introduce modified versions of the protein inside of the cells, you can do things like see if the proteins are near each other inside of cells with microscopy and various techniques. In vitro, you can do things like attach them to chips and plates and all this stuff. And so lots of cool stuff that you might see with Fry. So we often take advantage of radioactivity in biochemistry. Yes, even these days. If you think about elements, an element is defined by the number of protons, but the number of electrons can vary, which will impact the charge, and the number of neutrons can vary, and that won't impact the charge, but it might impact the stability of the nucleus. And if you make the nucleus too unstable, then you can end up with radioactivity where particles of energy are kind of given off. And we can measure this radiation that's given off as the molecules decay. If we think about the sorts of biomolecules and their elements that we see in biochemistry, and we want to see, okay, how can we uniquely measure DNA or RNA, or how can we uniquely measure protein? We often use P32 if we want to label DNA or RNA. So P, phosphorus. So if we think about the backbone of DNA or RNA, they've got those phosphate groups. We can introduce radioactive versions of that phosphorus in order to create radioactive probes of DNA or RNA. And one of the great things about these two is that they act very, very similar, almost indistinguishable from the normal one. So they don't get in the way of the normal reactions or normal processes, interrupt with things like you might find if you attached a big bulky fluorophore. So the benefit, or one benefit of the radioactivity, is that it is kind of unobtrusive and lets you see what's going on. A downside is that then you have to be working with radioactivity, and although it can be done very safely, there is a lot of expense, and it's a lot of work and paperwork and all sorts of things. And yeah, it takes a lot longer to do experiments involving radioactivity because you keep having to gear up and gear down and check with the Geiger counter and all this stuff. So I did a lot of work with radioactivity in grad school, and it was very, very sensitive. So that's another benefit of radioactivity. But yeah, some of the equipment is really, really old. It was funny. We had this one machine, and when it turned from 2020 to 2021, there was like a glitch in the software, and so it couldn't go past 2020, and so it reverted back to 1921. So that was a bit of a kick when I saw that on the screen as I'm like trying to finish my doctoral thesis work, and the computer goes back a decade. But anyway, yeah, it also had this like printer that printed on this dot paper, and the computer couldn't save or anything. It was interesting, but it was fun. And thanks to Dr. Nick Tongs for letting me use his scintillation counter. But anyway, so the P32, that's really good for DNA and RNA, and what I was actually using it for. So I use it sometimes for DNA and RNA with tracking things in like gels like these. But you can also use it for kinase assays. So a kinase will add a phosphate group from ATP onto a protein or some other molecule. And then you can use radioactive ATP to add a radioactive phosphate to see when the protein got phosphorylated. Okay, so that works great for DNA and RNA, but what about proteins? So proteins don't have phosphorus, but they do, well, some of them have sulfur. And DNA and RNA don't have sulfur. Only two of the amino acids have sulfur, so that'd be your cysteine and your methionine. And what can happen is that if you give them radio-labeled cysteine and methionine, where the sulfur is actually S35, then you can get the proteins to be labeled. So you grow cells in the absence of cysteine and methionine, and then you give, so the media does not have those amino acids, and you give them the amino acids, the radio-labeled versions, and then you could label the proteins. And what's really cool is that you can label it at different points. So if you don't give them cysteine or methionine, then you give them the radio-labeled versions, you can see the new protein that gets made as opposed to just all the protein that was there. So S35 is often used for proteins, and that's your radioactive sulfur. P32 is often used for DNA or RNA, or if you want to label phosphate groups for kinase assays and things, and so that would be your radioactive phosphorus. Not all of these isotopes, so when we talk about an isotope, that's a version of an element that has a different number of neutrons. Not all of the isotopes for an element are radioactive, so some of them are just heavy, and so like H2, so deuterium, or N15, or C13, these are molecules, these are isotopes, so they're the elements, but they act a lot like the elements, they act mostly almost like the elements, but they have a different number of neutrons, but not enough to kind of make things unstable, but enough to make things so that you can detect a difference. So this can be used to, say, track the metabolism of molecules. So you can use mass spectrometry, which will measure the mass to charge ratio of molecules. And if those molecules have isotopes incorporated that are heavier, then the molecules will be heavier. And so you could track the metabolism of different molecules. So all that metabolic tracking will be done inside of cells, but now let's get back to what we can do outside of cells. So if we have that purified protein, one thing that we could do is assess the stability of the protein. And so you might see protein denaturation assays, and this can be done with heat, or they could be done with chemicals like urea. And the basic idea is that the more stable the protein is, the more tightly folded it is, the more interactions there are holding it together in that 3D shape. So not the chain itself, but the actual folding of the chain. If those interactions are strong, then it's going to be harder to unfold or denature the protein. So when we say denature, that's talking about unfolding it. But if you give it enough heat, when you're giving it heat, it's allowing it to have the energy it needs to wiggle. And if it wiggles enough, it can break free of its folding, even if it's really sturdy. But it's going to take a lot more wiggling in order to unfold if it is sturdier protein. Similarly, it's going to take more chemicals that kind of disrupt things if you have a more sturdy protein. It's not exactly like this, but you can think of it kind of like how much soap you need to declog something that's really stuck together. And so the more soap it takes, the more chemical it takes to unfold the protein, to the sturdier the protein was, or the more heat it takes, the sturdier the protein was. So the more urea it takes, or the higher the heat you had to get to for it to unfold, will tell you that the protein was more stable. And so the way that we kind of compare these is with at least for the thermal shifts, are with heat denaturation at this value called Tm. And so Tm is the temperature at which like half of it is unfolded. And one way that we can detect the unfolding is by taking advantage of fluorescent dyes that bind to hydrophobic regions of proteins. Those hydrophobic regions are typically going to be hidden in the inside of the protein, away from the water. But when the protein unfolds, then they come into contact with the liquid, and then the liquid contains the dye, and the dye binds to the protein and fluoresces. And so you measure the fluorescence to see when the protein unfolded, and the more heat it took to get to see that fluorescence, the sturdier the protein. You could also think about doing something very similar if you have a concentration, say, of urea, so a denaturing chemical. You add the chemical. When the protein unfolds, you have some way to measure it, and so it could be with some sort of dye. It could also be taking advantage of things like intrinsic tryptophan fluorescence. So the amino acid tryptophan is naturally fluorescent without a fluorophore or anything. And so you can measure that fluorescence in order to see differences in the conformation or shape of a protein, if the protein has those tryptophans. So that will give you kind of like a general idea about the structural stability, but what if you want to go and actually look more at the structure itself? So now we're getting into structural biology, where we investigate the relationship between the form and the function of a molecule. So one thing is to look at its structure. So this can be done with techniques like X-ray crystallography, cryo-EM, NMR. Then see, okay, well, what's it supposed to do and how can we measure that? So we're testing its function, things like binding assays or activity assays. So we talk about an activity assay. That's basically if we have an enzyme and we want to see, can the enzyme do what it's supposed to do? Can we use the structure to predict changes that we can make to change the functioning, or see if for certain parts of the protein are really important for functioning? So then we can use site-directed mutagenesis to make specific changes. We can do truncations, where we remove parts of a protein to see, is that part important for this? And then go and see whether it can still do the things that it was supposed to do, and if it can do new things, and all these various cool stuff. So cryo-EM and X-ray crystallography will give you high resolution data. And so you're going to get data for where the different atoms are in the molecule. And so you don't see the atoms directly, but you get evidence of where they were through the evidence of the electrons and things that were in the atoms. And so it's a little complicated, but you end up with this sort of meshy looking thing called a map. and then you build the atomic model into that map. So you get this meshy thing, and then you say, okay, there's an atom here, here, I know the sequence, so this is how the structure is aligned inside of that map to best fit it, and this will be your experimental model. The finer the mesh of the map, the higher the resolution of the data, the more confidently you could actually place the atoms inside of that molecule. When we talk about a high resolution structure, it's a little confusing because it corresponds to a smaller number, because the resolution is the distance between two things that you can still tell are two different things. And so say you have dots on the wall across the room, and you have a pair of dots that's 1 centimeter apart, a pair of dots that's 10 centimeters apart, and a pair of dots that's a meter apart. And so, that would be 1,000 centimeters apart. And if you can tell the ones that are 1,000 centimeters apart are 1,000 centimeters apart, that's great. But if that's the closest distance you can make out, then you're not going to be able to tell that the ones that are 10 centimeters apart are actually 2 dots, or the ones that are 1 centimeters apart are actually 2 dots. If you had a resolution of 10 centimeters, well, now you could tell that the 10 centimeter ones were 2 dots, and you could tell that the 1 meter apart ones were 2 dots, but you couldn't tell that the 1 centimeter apart ones were 2 dots. And so if you had 1 centimeter resolution, you could tell that the 1 centimeter dots were 2 separate dots. And similarly, if you had like a 0.5 angstrom resolution, you could tell that 2 dots that were 0.5 angstroms apart. And so an angstrom is a 0.1 nanometer. And so that would be 0.05 nanometers apart. You could tell that those were 2 separate things, 2 separate atoms, say. Whereas if you had a 5 angstrom resolution, then you couldn't tell that those were 2 different things. They kind of just blur together. So lower number is higher resolution is better. And the better it is, the better your map is, the more confidently you can build that model in. So we get this data from techniques like X-ray crystallography and cryo-EM. And so X-ray crystallography, I did some of this in grad school, and it takes advantage of X-ray diffraction. So you get your protein to form these crystals, and this is often a lot harder than it sounds. You have to screen a lot of different conditions to get these proteins to crystallize. Once you do, then you shine X-rays at them, and the X-rays interact with the atoms and with the electrons in those atoms specifically, and that causes the X-ray waves to kind of interfere with one another in a way that gives you this pattern of spots. We call this pattern of spots a diffraction pattern, and you can actually, or computers these days, can interpret that pattern of spots and build backwards to give you one of those maps that then you can build a model into. And so this was just a home source. So this was actually at my grad school down the hall for me. We had this little X-ray machine. But when you want to go power and get high resolution data, you often go to a synchrotron. And so these are really big circular facilities, so big that they're like these tricycles that you use to get around. This is Brookhaven National Laboratory, BNL. And then they have these big X-ray machines, and you have robots that actually are controlling things. So it's a lot of fun, and I highly encourage you to get to a synchrotron sometime in your life if you have the opportunity. Cryo-EM. So cryo-EM used to be called blobology because you got really low resolution data, but the technology has improved a lot, and so it's coming really far. And so basically with cryo-EM, rather than using diffraction data where you're looking at the indirect evidence from the waves bouncing off of the electrons in the atoms, here it's more like a CT scan or a CAT scan, and it's taking these images of these kind of lots and lots of blurry images and then averaging the blurry images together in order to figure out this 3D structure. And so unlike with crystallography, the molecules don't have to like freeze in place. So you do freeze them, but you freeze them as they are. And so they're all in different conformations. They're all in different shapes and orientations. And then what happens is that you can kind of pick the ones that look similar and average the signals from those ones that look similar and figure out the 3D structure. So this is a very computationally heavy technique, and it is a very powerful technique. It works best typically for big complexes. So X-ray crystallography, everything kind of has to get, all the molecules have to crystallize, which means they kind of have to freeze in the same form, same conformation. Which with cryo-EM, here you are good for like complexes and things that maybe have multiple proteins interacting and they're not always in the exact same shape and they don't want to stay still. And so cryo-EM can be really good for that. it's definitely not just plug and go. There's a lot of optimization, and often you're going to have, if regions are too flexible, no matter what technique you're trying to look at them with, they're going to not show up on your images because they kind of just blur each other out. NMR can also be used for higher resolution data, but that's typically better for smaller proteins. And that works like an MRI. Those are more high resolution techniques, but there are also lower resolution structural biology techniques. So circular dichroism, or CD. CD will tell you about the secondary structure of a protein. So it's general overall, how much of it is alpha helix, how much of it is beta sheets, how much of it is unstructured. Those different types of secondary structure will interact differently with light. And so it involves the polarization of light and the rotation of light. And so I'm not going to go into the physics, but the key thing is that there are characteristic signals that you'll see from different types of secondary structures. And a protein, then you can deduce the overall composition of the protein in terms of what proportion of it is helical versus beta sheet. You can do things like you can actually use CD in combination with a chemical denaturant or in combination with heat to see how much heat or how much chemical it takes to get to the protein to unfold, where you would see the loss of those secondary structural signals. Or you could see under different conditions, does the secondary structure change? And so this is a much simpler technique, but a much less powerful technique. But depending on the questions you're trying to ask, it can be a great tool. And it can ask and answer a lot of questions that you couldn't do with something like X-ray crystallography or cryo-EM, where you're not going to there be doing things like the thermal denaturation. Another lower resolution structural biology technique is HGXMS, or hydrogen-deuterium exchange mass spectrometry. And it takes advantage of those isotopes we were talking about earlier. Specifically, it's going to use deuterium, and deuterium is a version of hydrogen that has a neutron. So if we think about protein, that is our normal hydrogen. It only has a proton. It does not have any neutrons. If you have a neutron, well, now you have deuterium. And so this would be our H2. And deuterium, so we call it, we have two because it has one proton and one neutron. And so H2, deuterium, it's not radioactive, but it is heavy. And so if it's incorporated into a protein, that protein will get heavier. And where can we incorporate it? Well, we incorporate it into the protein backbone. So in the protein backbone, we have those peptide bonds, those amide bonds that have this hydrogen on the nitrogen. And these can actually get swapped out in the solution. And so if you have a solution of deuterated water, so the water has deuterium, it can swap out with the hydrogens, the protium, in the backbone of the protein. And then that could label the protein. But that backbone is going to be tied up in secondary structure. That hydrogen is involved in hydrogen bonding a lot of the time. And so only if it's not involved in that hydrogen bonding, or only if that hydrogen bonding temporarily like breathes or comes apart, can you actually get the swappage there. So that's going to tell you about the secondary structure and solvent accessibility. So you have a protein, you dilute it in the deuterated water, and then the regions that are solvent exposed, so they're exposed to the deuterated water, and they're not tied up in secondary structure, are going to get labeled. Then you stop the labeling and you cut up the protein and do that mass spectrometry we were talking about earlier. And so the observable exchange is going to come from those backbones. And so the ones in the side chains are going to exchange back too quickly when you go to actually measure things. And the ones in the carbons are going to not be exchangeable because it would be too slow. And so what's going to happen is that over time, you're going to label more and more of the protein depending on its secondary structure and solvent accessibility. And regions of the protein that were bound to something will then be protected as well. And then you can go and you can cut up and measure the pieces and compare them under different conditions. You look over time, you take time points, and something that is flexible, something that is solvent exposed, something that has a not a defined secondary structure is going to take up a lot of deuterium really quickly. And so you'll see a signal that kind of like takes things up really quickly, as opposed to something that is not taking up the deuterium. And so that would be a more protected region. And so you might see things shown in like a spectrum, or you might see uptake plots over time, and you can compare different conditions. And so this is actually from the paper that came out of my grad school work. And so you can see that here, if something is red. So this is kind of this protein. We have this binding pocket. So this is this protein Argonaut, and it binds to this RNA. And you can see that the regions that are really dark blue, these are the regions that had really low uptake. And so you can see that these regions were really, really well protected. And so you can see that that's actually where that not only do they have a high secondary structure, but they're actually bound onto the RNA. So they're really well protected. But if the RNA is not around, then you're going to get a high uptake. The bottom line is that the higher the amount of secondary structure, the less uptake you're going to get. And the lower the solvent accessibility, the less uptake you're going to get. And if regions are more flexible, regions are unfolded, regions are dynamic, those regions are going to take up a lot of deuterium really quickly. And you can use this to analyze changes in the protein under different conditions. That's just a whirlwind tour of techniques, and there's likely many more you'll come across, or you'll never come across some of these in your lifetime. But hopefully this gave you an idea of the sort of techniques that are out there and being used by scientists that you might come across or not come across, or if you are interested in, you can look further into.